doing this, so I'm going to be pressing record, and I'll be capturing the most important stuff anyway. So I guess as to keep in mind on that topic is when discussing, or asking questions, try and have that higher level sort of, what did we achieve? What key knowledge gaps are next? And I guess briefly looking at tomorrow's agenda, we are running through more salmon specific related topics such as acoustics, genetic stock ID, eDNA and pathogens. That'll touch on some non-salmonid pieces as well, otoliths and scales, blood insulin like growth factor, food webs, salmon diets. You realize I've put that in twice there, so this would actually be salmon energetics. And in the agenda that I sent out yesterday, I had Charlie Waters from NOAA giving a presentation here, and I've taken him out as he is not available, so apologies for that mix-up. And then Mark Saunders, who was the International Review of the Salmon Director, will be closing out the workshop with a sort of IWS wrap-up and basin events for coastal impacts. Becky, a UN Decade of Ocean Science endorsed project that is being administered by Pisces, an update on that. So in terms of housekeeping, we're not in the Zoom webinar format like we were last time. So please raise your hand, your virtual hand, if you have questions. And I'd encourage you to keep your camera off unless asking questions. But if you do ask a question, it'd be nice to see you. And it'd be nice to see you presenting as well. So I am going to stop sharing my screen here. And just as a final note, as I will hand off to Brett Johnson here shortly. Just wanted to thank everyone too for being part of this project over the last 5 years really, and acknowledging that it's been something that's off the side of everyone's desks really, in addition to heavy workloads in academia and from your various agencies. And just wanted to send my appreciation to you for putting up with my sort of cat herding, nagging emails, as well as efforts from previous coordinators of IYS like Caroline Graham, Stephanie Taylor, Laura Tessier, Paul Kitching, Paulina Orlov, Mackenzie Kermode, and I'm sure I missed some there. But anyway, just a big thanks to everyone for being involved. So with that, I'm going to pass it over to Brett Johnson, who will be speaking to an update on data mobilization efforts.
Thanks, Aidan. Okay, so I'm just going to go over this briefly, this International Year of the Salmon data mobilization work that we've been involved with since 2019. My name is Brett Johnson. I'm from the Hackeye Institute, and this work was done a lot with Tim Vanderstapp as well, who was not able to make it today. So I am going to give a brief background. We do have a bit of a varied audience. I'll keep that short and then talk a little bit about what our objectives of this data mobilization project were, talk a little bit about the services we provided and what infrastructure we built, and then kind of go into the impact of the IYS data so far, and really just try to focus on the last point a little bit more in terms of the IYS legacy plan and what our next steps are going to be. So yeah, brief background. In 2019, Mark Saunders came to us at the Hackeye Institute, looking to get some help on mobilizing the data and managing the data from these big expeditions that were coming. So we agreed to do that, and we're quite connected with the Canadian Integrated Ocean Observing System, as well as the Ocean Decade the Ocean Decade Collaborative Center for the Northeast Pacific, which is the UN Decade of Ocean Science Collaborative Center. And so it's kind of a natural fit for us, given the international nature of the work. We aligned our work with the Canadian Integrated Ocean Observing System, but also the Global Ocean Observing System of the United Nations. And together with the IYS and the NPAFC, we embarked on this salmon data mobilization project as a partnership, essentially. And our objectives were really to design a data mobilization process and infrastructure using internationally adopted best practices to standardize, integrate, and share the data. And that was not only to share the data within the expedition participants, but also to deepen the impact of the IYS by making sure these data were available publicly where appropriate in perpetuity. So to do that, we had to build the necessary data cataloging and data hosting infrastructure while aligning the standards with the Global Ocean Observing System. And so in terms of the services offered, we developed a strategic plan for data mobilization in collaboration with the IYS Secretariat and the chief scientist. We also developed a data policy, a data sharing policy, and we built a data template to try to standardize the sharing of data, especially the trawl data, coming in advance of the 2022 expedition. We collected and curated a lot of metadata and published a ton of metadata and actual data, one for global integration and access, but also within the project to make sure that internal access was facilitated. And to do that, we built a data catalog and IYS data mobilization portal, which was kind of a one-stop shop for what to do with your data and best practices and data summaries, et cetera. And we did that all using a federated metadata and data architecture. So really what that means is that the metadata that we were producing could be harvested by a number of different indexing search engines such as Google or other catalogs to really make the IYS data discoverable across a wide variety of platforms. And the data architecture was also federated in that we published data to domain-specific repositories, depending on the scientific domain and where the appropriate repository was for that type of data. And so probably two of the most important pieces of infrastructure that we built were the IYS data catalog. That's available at iys.haki.org. And it will always be available at this URL moving forward. And we also built this data mobilization portal. And so those are the URLs. I encourage you to check those out. These are also QR codes. So if you take a screenshot, you'll be able to click those icons. And so just a little bit about the impact of the IYS data so far. We've managed to publish 50 data sets. I think 33 of those at this point are publicly available, and the 16 of those are internally available. We hope to make those public, more on that in a little bit. In total, we've got about 148 data set citation, and there's been almost 25,000 downloads of the IYS data sets. And we've got 82 metadata records published in our catalog and 118 connected scientists. So that's the number of scientists that were involved with the production of all these different data sets. And so that was pretty cool. This number is just going to continue. These numbers are going to continue to go up in terms of downloads and citations. So it'll be interesting to track that over time. And in terms of the infrastructure, this is just a diagram of kind of what I was talking about in terms of the IYS data catalog being integrated with a variety of different search engines and other catalogs. And as I was saying, the IYS data, all of the raw data that we received, we published it in a GitHub repository, where then we would transform the data and publish it in a standard data repository, such as the Ocean Biodiversity Information System, or for physical and biogeochemical data, we would put that on an ERDAP data server following the climate forecast conventions. And so one of the most important data products that we were able to bring together was this integrated data collection, which is really an integration of all the different trawl and catch data sets from 2019, 20, and 2022, as well as the zooplankton bongo data and the CTD data at this point. We would like to incorporate more data as it becomes available, and we'll plan to do that over the coming months. And at this point, this integrated data collection is available internally only, as per the IYS data policy. We hope and plan to make that available on March 31st, more broadly and publicly. But up until that point, the IYS data policy applies to using those data. And if you need to get permissions to access that data, please see the data mobilization portal at this URL for instructions about how to do that. And so really the next steps, if you have not sent us data yet, please do. I know a lot of you are continuing to work on standardizing data sets and bringing it all together or doing laboratory analysis. So yeah, as those become available, please submit them to our email address, which is iys@hacki.org and secretariat@npafc.org. Please just submit the data with some sort of data dictionary describing the data columns and a README file, which should provide some indication of the protocols and limits to interpretation. Again, the data submission best practices are on the IYS data mobilization portal. Next steps continued. We're going to continue to receive and publish new data. We'll also update any existing data sets with any new versions as required. So if you've been working with some data and you've made some corrections or noticed some errors, please pass that information on to myself or the Secretariat, and we can make sure that the newest versions of those data sets are made available. Lastly, Tim is spearheading a data descriptor paper for the integrated data collection. So that we'll be targeting a scientific data journal for that, which is really just a description of the integrated data collection to really facilitate reuse. So if you are one of the chief scientists on those vessels, look out for a draft in your inbox, and be great to work with you on that moving forward. So more to follow on that. And then lastly, we're working with the NPAFC Secretariat, specifically William Stanbury, the Web and Publications Manager, to really manage the IYS legacy. So we're training Will to continue on with the work that we've been doing. And so a little bit more on that, the IYS legacy plan. As I've mentioned, most of the data will be made publicly available on March 31st, 2024, as per the IYS data sharing policy. Of course, there will be some reasons to embargo data beyond that if students are needing those data sets for their publications, for their theses, etc. So we'll be in contact with all the data sets that are currently internally available. We'll be in contact with the data providers of those data sets before we make them public to make sure that's okay. And then otherwise, Will is going to continue to accept and publish data when our contract ends, which is March 31st, 2024. And beyond that, the real legacy is, the IYS catalog will essentially transition to becoming the NPAFC catalog. And yeah, we're excited about that. Will's excited, I think, about taking that on. And now the NPAFC has this kind of world-class data infrastructure for data and metadata sharing. And as far as I understand, they're planning to continue to host important data sets from future cruises and also transition to using the or the IYS infrastructure for NPAFC data sets that support their regular reporting requirements. So I won't go into too much more detail other than just to ask that when people are publishing manuscripts using IYS data, please include the DOI for your data set in the data availability statement of your manuscript. So your DOI, you can find that in the IYS data catalog. And then if you're citing other IYS data sets in your paper, please cite those data sets formally in text as well as in the list of references. This just really helps with reproducibility and findability and reusability of the IYS data set to really deepen the impact of all the amazing work that everybody's done over the last few years. So I do want to take some time for questions. I know that was kind of a broad overview. So yeah, I will also pop in the chat here If you don't feel that there's time to ask a lot of questions here, we've got this little Slido poll. So please click that link and you just paste in that code. And there's just a one prompt question that we would love to get your feedback on what worked and what didn't work for you, the IYS participants, in terms of data mobilization, and as well as thinking about what we can focus on in the next 6 months to deliver the requirements for you guys. So with that, I'll take any questions. Thank you.
Thanks very much, Brad. Wanting to second or highlight the strength and great support that the IWS project has had from the Hacker Institute over the last 3, 4, and even more years. And to highlight that, you know, once we've all wrapped up here today and go back to our own projects, the legacy via the data and the availability of the data will be the legacy, really. So highlighting the breadth asked there for folks to submit data to via the relevant avenues here that Brett's spoken to, ideally by March, and if not, when it's ready beyond that. Any questions for Brett? Also pop them through the slider there. I know we got one via email, Brett, from Suzanne, just about submitting and metadata. So perhaps you can take a look at that one.
Sure.
Okay. Feel free to pop any questions in the chat if they come up after this, but I think we'll move on to our next presentation from Laurie Whitecamp, giving us an overview on trawl catches from the expeditions.
Great. Can you hear me okay?
I can hear you.
Excellent.
And I can see your presentation. Great.
Perfect. I'm going to turn my camera off just to ensure I've got full visibility. So what I wanted to do is just provide a quick summary of the catches by the trawls, which was the main form of fishing that happened out there. And I show you the study area again, just to remind you more than anything that the Northwest Explorer and the Tinro were in the western part of the study area. The Shimada was kind of in the middle. And then we had the Franklin, which was using a trawl, and the Ross Spirit, which was using other methods, primarily long lines and a gill net in the eastern part of the study area. So the Franklin, the Shimada, and the Ross Spirit are the ones that really overlapped the 2019 and 2020 study areas. And that we did this common methodology across all 3 ships. So we all did a full suite of physical oceanography, biological oceanography, and then we fished. And most of the ships used trawls. The type of trawl varied a little bit, but they're all large rope trawls towed to the surface. And then the Ross Spirit was using gill nets and long lines. Just effort, it really varied on when people were out there. So the Shimada, shown in white here, was out there first. We did our first set, I think, on February 5th. You can see the peak of activity in late February, early March, when we had 3 ships out there. And then the Northwest Explorer was the last one. They were the only ones out there in April, and we were lucky to have them out there at all. So even though there's not complete overlap in time, certainly in space. So this is just a summary of the effort. Most of the ships were able to do between 21 and 34 sets, completed trawls. Most of them were during the day, but particularly Shimada and Tinro, almost half of them were at night. And then this is the catch numbers. So this excludes jellyfish. Jellyfish tend to break apart in the net, so you don't know how many you've got. So we just weighed those, and then the total catch by ship. So this is photographs just showing all the catches on the Shimada. I think we made 21 catch separate trawls that were successful. And you can see the variety of salmon. All this white stuff is jellyfish. There's some squid in there, miscellaneous other stuff. I don't have a picture of the shark we caught, so it's not on this, but just a diversity of organisms out there. This is by taxonomic group. So you can see we had 55 identified taxonomic groups, 42,000 organisms, which excludes the jellyfish, and about 3,000 kilograms of stuff, which was mainly composed of salmon and jellyfish. Those were by weight the biggest two we saw. This is just showing the frequency of occurrence of all the different species by group. So obviously some salmon, jellyfish were pretty common. Some of the squids were pretty common. And blue lanternfish or Tarlingtonia were also very common, caught a lot. And also you can see that some things are caught more at night than during the day. For example, coho was the only salmon that we caught in larger numbers at night than we did at day. Certainly most of the squids and the myctophids also, and as well as other fish, actually, most of the other fish, we also caught more of them at night than we did at day, during the day. So really dramatic differences, depending on what time of day you're fishing at. This is just showing the distribution of the different salmon species that are caught in the trawls. So this is counts per hour in the trawls. I'm not showing the longline or gillnet. And sockeye was our #1 species, but I think it's the circle with the red ring around it is where about half of the sockeye were caught. The tin roe caught over 600 sockeye in a single set. And you can see that obviously Chinook were a lot less abundant. And there's also differences north-south, but not really big ones east-west as far as where the salmon are caught. And I don't have steelhead on here because none of the trawls caught steelhead, but that was the #1 species of salmon caught by the gillnet longliner on the raw spirit. So very different catches, depending on what kind of gear you're doing. And we know steelhead are very surface oriented, and the fact that we didn't get any in the trawls. And I hope Christoph will talk a little bit about that with eDNA. It sounded like there's evidence that they were out there and we just missed them with the trawls. If you put the counts by station for the trawls into a MDS plot, you can see there's almost complete overlap in MDS space. We really didn't see big east-west differences. We did see some north-south differences. And then these are the correlation coefficients, again, based on this counts by station. You can see pinks, chum, and sockeye are all highly correlated with each other. Chinook and coho much less so compared to other species. So some interesting patterns. And I think this really reflects kind of that north-south distribution of salmon. If you add the gillnetter into it, where again, the steelhead were their #1 catch, it's interesting that the steelhead were kind of weakly positively correlated with pink salmon and fairly strongly correlated with coho salmon, and then negatively correlated with the others. And I haven't seen the map showing steelhead catches compared to the other species, but it's kind of interesting, as in whether it's really south or north or whatever. And if you run the MDS again with all 5 boats on it, the catches of all 5 boats, the pink squares here are the raw spirit, and they really still kind of fall into more or less the same space, even though they were catching steelhead. It's 1 species out of 6, I guess, that were out there. So didn't have a huge impact on the MDS patterns. It's interesting too, if you look at the catch at IYS across all species, you can see, obviously we caught more sockeye than anything else. And I thought it was interesting to compare that to the catch across the entire Pacific. This is commercial catch data from NPFC. And you can see the dominance of pink salmon, basin-wide across the entire North Pacific compared to what we did, where pinks made up a tiny little bit of it, fraction of it. And then you can also just look at US and Canada catch and see the same thing. So, and I do the US and Canada just because the Russians tend to catch a lot of pinks and chum. So in both cases, you can see some real differences between what we caught and what, commercial fisheries are catching out there. And we can also compare the catches that we saw. And here I've just reduced it down to the eastern part of the study area. So catches by the Franklin and the Shimada in 2022 compared to what we saw in 19 and 20 in the Gulf, where in those cases, chum were much higher than we saw in 2022, where sockeye were really dominating. So some interesting patterns going on. We also got a lot of other stuff. This is just kind of showing distributions of some of the other major species. So we've got 2 different species of squid in the leftmost column. I've got 2 species of lanternfish or myctophids in the center one, and then the bottom center and the right column are all the 4 species of jellyfish that we caught a lot of. And I think in some ways the important thing is just that we didn't see big east-west differences in catches at all. What we were getting in the western part of the study area was the same as what we get in the eastern part, which I think is really fascinating. I had expected that we might see some differences as you move further out, given that we were sampling over such a large area, 2.5 million square kilometers. And to kind of back that up, if you do the MDS plots, these are all species, including salmon, and those main ones that I just showed you. If you look by count, which excludes jellyfish, or by weight, which includes jellyfish, there's really complete overlap, more or less, in this MDS space. Maybe a little bit of a difference, and here I'm using ship location as a proxy for geography or location across the study area. So we really didn't see big differences by area, which I thought was really interesting and not something that I expected we would see. And that's all I've got. So I will stop sharing and open it up if anybody has any questions. And this was really just intended as an overview. I don't expect any questions, honestly. If we've got questions on this, then we've got problems. So.
Thanks, Laurie. You managed to just squeeze a lot in there and added some new things too, I think, from when you presented in June. So really appreciate that. I was really fascinated by the pie graph you made comparing IYS catches to commercial catches in the North, the East and Western Pacific. Would you mind bringing that up again, actually, if that's not too much of a bother? Just interested if you or others have any thoughts on why, on the differences there. With pinks dominating.
I think Skip's going to talk to that, and it really seems like we didn't go far enough south. I think is based on the historic data. That's what it points to. But at the same time, Josh, now I'm forgetting his. Josh from the University of Alaska did a model based on temperature and the historic data, and he showed that there were areas where we should have caught pinks, or the tin row should have caught pink salmon, and they did not. If you take the historic data and kind of this temperature template that goes with it, and it's really puzzling, what was going on. One part of the equation, and maybe the genetics on the pinks aren't fabulous. We can't really resolve, basin specific differences in pink salmon like we can some of the other species. But where's I going with that? The catches in Russia in 2022 were much lower than they had been. They've been having some enormous catches, off the charts, like highest ever. I want to say 450 million pinks were caught in 19 or 20, a couple of those years. Anyway, 2022 was not a big pink year in Russia. So maybe part of this reflects the overall lower abundance of pinks across the North Pacific. And I hope when Skip gives his talk on the historic data, he can talk a little bit more about that. But it's really puzzling. Where were the pinks? Especially if you think about, Greg Ruggiero and company's many papers that are kind of blaming pinks for everything. All these interactions, reduced productivity. We're not seeing it out here. The opportunity for interaction does not look very high from what our data show. So, which is interesting in itself.
Thanks, Laurie. Got a message from Skip saying that he will speak to this in the live presentation spot later today. So a good hook to keep you around for that live presentation. But while we got a minute or two, Skip, do you have any sort of brief comments while we've got those figures from Laurie fresh in our minds here?
Stay tuned for my talk. but I there is a possible reason.
Okay, intriguing, a great trailer for what's to come. Okay, unless there are any last minute questions, then you can always pop them in the chat and folks can respond. We're going to move on to plastics. And we're going to start off looking at myctophids and squid from Suzanne Brander, Oregon State University. Take it away, Suzanne.
All right, just a moment. I'm going to go ahead and share my screen. All right.
Looks great.
Wonderful. So I'm going to be talking about the presence of microparticles in myctophids and squid. So kind of moving along a little bit from salmon, but then I think there'll be another presentation following mine on microplastics in salmonids. So this was work that was completed in my lab, but we were pulled into this project, thankfully, by Laurie Whitecamp, who just presented. So really glad to have been able to collaborate with her on this work. So it's a collaboration between Oregon State University and the Northwest Fisheries Science Center here, both at the Hatfield Marine Science Center in Newport, Oregon. So just to kind of kick things off, and if you've been following the news or the literature, you know that there has been a lot of information, a lot of papers published recently on microplastics occurrence in the ocean. We know, of course, now that it is a problem that extends far beyond the ocean, that we find plastic particles and pollution in freshwater, in terrestrial areas. It's even now in atmospheric circulation. But this graph gives you an idea of what the different sources are. As far as ocean microplastics, we know that synthetic textiles now appear to comprise the largest percentage of particles that are being found in the oceans. Our research findings support this, and I'll talk a little bit about this in the few minutes I have today. So what are microplastics? They are defined by the state of California now, which published one of the first definitions as solid polymeric materials to which chemical additives or other substances have been added. And they are particles which have at least two dimensions that are greater than 1 micron and less than 5 millimeters. Part of the reason for that 1 micron cutoff is that as of today, it's not possible in most cases to detect particles that are smaller than this, even though we know that they're out in the environment. We know of our typical plastics that we see and try to figure out if we can recycle or not. Unfortunately, many of them we can't. And again, some of the main sources to the ocean and to waterways more generally are tire particles and textiles. And fibers, of course, are ubiquitous and found basically everywhere we look. Our research extends beyond myctophids to other fish species, to zooplankton. We've looked in scat from different animals. We are also researching the impact of particles in fertilizers and composts, and really across all of those different environmental matrices. Fibers are usually at least half, if not more, of what we find in terms of particles, both synthetic and human-made, or natural but human-modified. So getting back to the Pacific Food Web and our focus for this study, we were really interested in looking at myctophids and organisms that prey upon myctophids. because of this vertical migration that they undertake on a daily basis. And we were wondering whether they are basically a vector that's transporting microplastics across these mesopelagic food webs. And so the two projects I'm going to cover today, one, of course, is the International Year of the Salmon funded project, where we looked at myctophids and squid caught from 3 of the cruises, the RV Tinro, SBMS, and the IYS Franklin. And then I'll talk a little bit about research that is related to this on some other projects we've recently completed on coastal and museum myctophid specimens. But just to kind of give you an overview of what our lab group does with samples like this, everything again from zooplankton to scat to whole fish samples and water samples, is that we isolate and dissect biota, or we digest samples depending on what they're comprised of. They're carefully inspected and individual particles are measured and photographed, and then particles, at least a percentage of them, are verified using Fourier transform infrared spectroscopy. So it's a lengthy and time-consuming process, but we are able to get accurate identification of particles at the end, and we're able to differentiate as to whether they're plastic, natural, or anthropogenically modified in some way. So this is a summary of some recent work that Olivia Boison, who was the master student who was helping with the IYS research as well, she was able to obtain myctophids from NOAA cruises that were conducted over the past couple of decades off the coasts of Washington and Oregon. And one of our big questions was whether we were seeing an influence as far as amount of microplastics or microparticles taken up by these fish, with if there was a connection to proximity to the mouth of the Columbia River. What she found is that there really wasn't. And most of these samples were taken between 15 and 30 miles off of the coast, so that might be why. But she found that about 34% of these 3 different myctophid species that were being sampled by NOAA contained particles, and the majority were fibers, as you can see from her pie charts here that are on the slide. So for this study, we found 142 suspected particles in 340 myctophids, which equates to about 34% having 1 or more particles. This is similar to other research findings from 2010. And the average size was close to 1 millimeter. And what we are sort of inferring from these findings is that myctophids are probably getting these particles from their prey. They're not ingesting them directly. And that's been seen in the lab too, that fish tend to consume more particles, especially fibers, if there's a trophic transfer step. And so we think that they're probably obtaining these particles from krill and other commonly ingested prey items. For museum specimens, Olivia was able to get specimens from 2 different locations, Scripps and the University of Washington. And we were able to look at myctophids from 5-year span starting in the 1960s. And she looked at 3 different species for this study as well. And what we found was a little bit surprising because we were expecting to see a greater and faster increase in the number of plastics being consumed by these fish. And we found that there was a slight significant trend with one of the collections, the Scripps collection, and overall about a 1.1% increase in particle uptake per year. And about 24% of those fish were found to contain microparticles. We think for both the NOAA study and this museum study that because the fish had been preserved for some time, there may have been some breakdown of the particles, because now I'm going to move into the IYS research where we did find slightly higher numbers of particles per fish in freshly caught specimens. So again, we received myctophids and squid, mostly small armhook squid, from 3 different cruises, the Shimada, the RV Tinro, and the Franklin. And our questions were, are squid and myctophids consuming microplastics? And we were pretty sure the answer to that was yes. But the more interesting questions were, is there a difference between those levels in the different species? What could be the drivers of that? And is there any evidence of bioaccumulation or biomagnification? And this is a summary of our results. So in the myctophid GI tracts, what we found was that there were some with no suspected plastics. So about 28 of those fish that we dissected didn't have any suspected plastics. 77 did. So sort of in alignment with other work that we've done. In the squid actually, a larger percentage of them contained suspected particles. And in terms of particle types, the majority of those particles were anthropogenic. When I say anthropogenic, I mean that they may not necessarily be plastic, but they were modified cotton. Often cotton is chemically treated in ways that make it persist in the environment. Sometimes as long as plastics do, we find those in the guts of organisms all the time. And we found that 24% of the particles found in myctophids from these cruises were synthetic, and 14% of the particles in squid were synthetic. And of course, this was a relatively small sample size, but we looked at 105 total myctophids and 41 of the squid. And so 73% of the myctophids, again, had fibers or particles, and we found an average of 1.6 suspected particles or plastic per myctophid. So a bit higher than, again, the preserved specimens that Olivia was examining, but within the range that others have found. In the squid, we found that 90% of those squid had fibers or particles, and the average number was nearly double what it was in the myctophids, so 3.9% particles on average. Is this evidence of bioaccumulation? Possibly, but we would need to process more samples really to tell definitively, but it's interesting to see that difference between these two different trophic levels. So what are the implications for fisheries? If you look at this on the surface, it doesn't seem like 1.6 or especially 0.42 particles per fish is really that big of a deal. But if you scale it up to what a predator might eat, a Pacific albacore, for example, will consume 1,600 metric tons of myctophids. And if you go with the lower estimate from our study of preserved specimens, 0.42 particles per a 2.23 gram myctophid, that comes out to greater than 300 million particles per year. And of course, it would be much higher if you assumed the particle count per fish was 1.6. And so this is a concern for Pacific albacore. It's also a concern for aquaculture, given that we are sometimes grinding up fish species like myctophids into pellet form to use for feed for aquaculture facilities and purposes. So, what can we do? If the big concern, or one of the bigger concern, is about fibers, there are ways of removing fibers from our laundry before that gray water leaves and is treated at wastewater treatment plants. And I'm actually a co-PI on a new study that is funded by Oregon Sea Grant that is going to look a bit further into this to see if doing a pilot installation of filters on washing machines in a couple of coastal towns in Oregon, if that causes a significant and measurable reduction in fibers being released in effluent or being released to coastal areas. So that work is starting up in 2024. So it's a frustrating problem, but there are potential mitigation strategies on the table. So I'll finish up there to make sure I have plenty of time for questions and for the other presentation on plastics. But just wanted to give a shout out to some of our other funding sources in addition to IOIS, mainly the Pacific Northwest Consortium on Plastics, which has been in effect since the end of 2019. And it's a group of us here at Oregon State and Western Washington University that are working on trying to better understand the toxicity of micro and nanoplastics and microfibers as well. So more information on our website there. And I also want to acknowledge the hard work of all the students and colleagues that were involved in the different projects I overviewed. Thank you for your time.
Thanks very much, Suzanne. I'm not seeing any hands up immediately. So what we might do is roll into Gennadi's plastics presentation, and then we can have questions on both presentations at the end. Oh, we got one from Rick.
Hello all, I will try to just start from my screen. Okay?
Thanks, Gennadi. Suzanne, hopefully you can see Rick's question in the chat there, so you can get back to him there. So it's, go ahead, Gennadi.
Do you see my screen?
You should be able to. Are you having trouble with it?
I will try again. Do you see my screen?
Brilliant. I see it loud and clear.
Okay. So very briefly, yes, just few slides about our results from plastics research, because we covered not only microplastics phenomena, but also macroplastics, trying to, in last expedition, to make any remote by GoPro observation. So go ahead, very brief intro. So the summary of this slide, very simple. Biota has a very strong sense in North Pacific ecosystems, and in scenario possible for just coming time and for the possible future, we have for oligotrophic region mostly high plastic. Probably in some point we should have the same, and it's waiting for North Pacific ecosystems. We should have downing the plastics concentration, because again, biota has very efficiency to remove plastics from, for example, in our region, and I mean North Pacifics. And what we have after years of our project, I mean International Year of Salmons. First of all, in after 19 expedition, we published results, but it's mainly regarding particles, no fibers, unfortunately. We also have possible good perspective for GoPro data, still not analyzing and just waiting for the time. And what I want to say now, what we confirmed and detected fibers and less than quantity of fibers plastic in the salmon. So question remaining about North Pacific ecosystems, because that high productive area with very seems to be effective with plastic removal. And very briefly, our results here, still unpublished. So for environmental, I mean, it's mountain, just surface Neston, it's trolling. Fibers everywhere, 100%, and particles less, about 50. We see proportional And again, fibers just prevailed. And in Gulf of Alaska vicinities, confirmed as region of dissipation of plastics. Again, already published at good point of that paper, Egert et al. Just compare Great Pacific Garbage Patch with North Pacific Aquatory with our data. And seems to be we have same preliminary result for Central Aleutian, I mean Southern Ocean part, not in the Bering Sea. For salmon, it's interesting that concentration, again, it's not quantitative data, less and more in no Chinook data enough for statistics, no still head data. And again, fibers prevailed totally, chemically confirmed, and all of that is anthropogenic, excepting some question remaining and needing consult chemical guys. Well, next steps and acknowledge, and as you see, well, we have good rest of remote sensing data, and GoPro should be embedded in our analysis just to understand what we have in plastic here, I mean in North Pacific after all our efforts. And other big question for Pacific subarctic, so is utilization facility for plastics, of course, not endless, and we should understand what is happening here. We should to better understand. And for me personally, have you not links between plastics and salmon, its population dynamics here in the open ocean? My warm words and acknowledge all crew involved in this job, planning, fulfillment, and field work for our leaders. I mean, Evgeny Pakhomov, Alexei Somov, Semyon Novokrechenych with his bright idea to just take a look inside salmons. Michael Kolobov from Moscow State University having big help as assistant with Maria Gavrilo, my co-authors. Of course, Ocean Cleanup team and GoPro teams. Well, it's all my talk for today. Thank you very much.
Thank you very much, Gennady. And thank you for your ongoing and continued passion and energy in the plastic side of things here. You've been a real force in driving that group forward, and often joining many meetings at very late times at night or early in the morning, like I'm sure it is at the moment too. Are there any questions for Gennady? You're muted, Gennady. I can't hear you. I'm sorry.
Okay, it's okay. Not so important. Glad to see all of us again.
I'm not seeing any questions. Feel free to put them in the chat for Gennady. But I think we are going to move on to Bernie Yang from University of Victoria. Speak to physical oceanography. Bernie, feel free to share screen and go ahead.
Okay. You'll be able to see it, I think.
It's loading on my end. Now I can.
Okay. Okay, thank you everyone for coming in. I just want to first talk a little bit about some of the work I've been doing with glider observations in eddy in the Northeast Pacific, and then I'll give you more of a brief overview of the sort of the spatial patterns of different physical properties and the mixed layer in the Northeast Pacific that could assist you to interpret some of your own results. So a, a large part of my project was to understand the sort of the structure and offshore transport of these eddies generated close to the British Columbia coast. So on the left-hand side, I'm just showing sort of a picture of it, and then the right-hand side, I'm showing you a map of the eddy. So there's one of these eddies that generated in 2018, and then it propagated all the way offshore. So that's being shown in the gray track over there. And we deployed a glider along line P, which is labeled over there. And then it intersected this eddy. So I'll just briefly talk about some of the interesting parts of the eddy, and then move on to the IYS expedition results. So what I'm showing you over here is the general circulation pattern in this Northeast Pacific region, and then I also labeled line P over there. But the key thing from this picture is that there are three sort of major currents. There's the Alaska current, the California current, and the Alaska gyre as well. And most of these currents are actually going eastward. So some of these offshore propagating eddies that are pretty much the only things that are going westward could be very important in transporting coastal waters towards the offshore regions, which could be important for salmon. And so what I'm showing you over here is just the sort of the cross section of one of these eddies that are glider sampled. So panel A is showing you the temperature, panel B is showing the salinity, and then finally panel C is showing the oxygen in this eddy. And really the key point is that these eddies are transporting water with higher temperature and then lower salinity, as well as lower oxygen waters from the coastal regions towards the offshore regions. And I think the most important part of these observations pertaining to salmon might be the nutrient properties in these eddies. So the glider also sampled like 4 key nutrients: the dissolved oxygen in panel A, and then the nitrate in panel B, phosphate in panel C, and then finally silicate in panel B. So on each of these panels, the red curves are some of the profiles sampled closer to the shore, while the blue curves are the profiles sampled offshore. So those are marked on the map on the right. And then finally, the black solid curve is the profile sampled within the eddy. And really the point is that the eddy actually carries lower oxygen water, which I talked about just on the previous slide, but also slightly higher nutrient water compared to the offshore region water. So you look at panels B, C, and D, you can see that especially near where the eddy core is at about 200 to 300 meters, you can see that there's elevated levels of nutrient compared to its surroundings. So these eddies that are generated near the coast could be something very important for the physical and the biological environment, because they carry coastal property water with higher nutrients towards the offshore. So that's really a very quick synopsis of some of the work I've been doing. But I just want to shift gears to talk a little bit about the general conditions of the Northeast Pacific. So what I'm plotting over here is the average, the mixed layer depth in winter over 20 years from Argo climatology data. So I think a lot of the mixed layer properties might be very interesting to the ecologists. And the key point from this is that the mixed layer depth has, the climatology has sort of a spatial gradient to it. So if you look in the offshore and onshore component, that's the right and left arrow, you can see that the mixed layer goes a little bit shallower near the shore, and then goes deeper at the offshore region, which is kind of what we expect. But there's also a sort of a northwest southeast gradient to this mixed layer depth. So if you look at the northeast part, the mixed layer depth tends to be very shallow, while if you move down towards the southeast part, the mixed layer depth tends to be a little bit deeper. And so what I'm showing you over here is the 2019 data of the properties inside the mixed layers. So panel A is showing the depth, panel B is showing you the temperature, and then finally panel C is showing you the salinity inside the mixed layer. And while the mixed layer depth doesn't have like a very significant spatial pattern, but you can see these northwest southeast gradients clearly inside the temperature and salinity graph. So in the mixed layer, there's lower temperature at the northwest part, and then there's higher temperature at the southeast part of the data set. Well, the salinity is kind of the opposite, where you have higher salinities at the northwest part, and then fresher water, so lower salinities at the southeastern part of the data set. And if we compare the data to the Argo climatology, you can see that the mixed layer depth is on average quite a bit deeper at most parts compared to the previous data. And then in terms of the temperature and salinity, so your salinity is panel B, and then temperature is panel C. You can see that in the data set, the salinity is lower on average compared to the climatology, while the temperature appears to be most of the time is a little bit higher than the climatology. And here's the same data for 2020. You have the mixed layer depth on the top left, and then the salinity on the top right, and then finally the mixed layer temperature in the bottom left. So in this year, I think there might be something about the sampling track or the sampling dates that could be affecting how the spatial variability shows up in this data set. But if we compare it to the Argo climatology, we can also see that the mixed layer depth is generally a little bit deeper than the climatology, while the temperature shows a little bit more variability. But you can also see that the salinity is lower compared to average in this 2020 year. And then this is the mixed layer depth in 2022. So over here, you can also see this northwest southeast gradient where the mixed layer depths tend to be a little bit shallower in the northwest parts, and then a little bit deeper in the southeast parts. There's also this same gradient happening in mixed layer temperature over this data set as well. But it doesn't really show up too much in salinity. And if we compare it to the climatology, it's, I think, really the same story as 2020, where the mixed layer depth tends to be a little bit deeper compared to the average. And then while the temperature is a little bit more variable, we can also see that the salinity is a little bit lower than on average. So what I think really the key point is that there's usually this very strong gradient between the northwest and southeast part of the data set. And also in most of these observational years, the mixed layer depth was a little bit deeper than average. The temperature is a little bit warmer than average, and the salinity is a little bit lower than average. So I think these are really some of the key points that the CTD data show. So that's what I have to say. If there's any questions, I can answer it.
Thanks very much, Bernie. Really appreciate you breaking that down and displaying the CTD data in depth. Any questions for Bernie? Now we've got Marta Connick on the call, who'll be speaking a little later. Oh, actually, we'll do that question from Laurie first. What causes the mixed layer depth to change?
It could be due to like wind-driven mixing, because the wind actually determines the mixed layer. So if you have stronger winds, the mixed layer depth might be a little bit deeper than usual. So that's one way the mixed layer depth can change. The other one is like mainly due to the temperature stratification at near the surface. So if you have a stronger temperature stratification, then mixed layer depth might be a little bit shallow. So that comes down to, you know, some of the surface mixing as well.
And generally speaking, would you say that the years that the expedition occurred were sort of normal, abnormal, or in between for mixed layer depth?
I wouldn't say they're abnormal. There's certainly like a trend, like I said, the deeper mixed layer and the salinity. But if you look at the temperature, it's kind of, some of it is a little bit higher than average, some of it's a little bit lower. So I don't think it might be like an abnormal 3 years. So it's just the 2020 data was a little bit, I would say this, because the sampling track is not very as structured as the other 2 years. So there might be some sort of time dependence of the data in there.
Right.
So some of the, you can see some of the data points. Maybe if I go back over here a little bit, like if you look at temperature, like some of the data points might be, there's a red and then might be an adjacent blue. So that might be because those two stations were actually sampled very far apart in terms of time.
Right. I see. Just to clarify, did you say that the 3 years were abnormal or normal?
I was in normal, not
normal. Correct, Roger that. Thanks. There's a question for you in the chat. Just for the sake of time, I might let you answer that in the chat, Bernie.
Okay.
We will move on to Richard Dixon from the University of Victoria.
Actually, Aidan, I think Emily Lamaghi had she was going to slide in here with a little bit of a presentation on some.
Oh, great. Yes, I knew she was sliding in. I just thought it was later, but absolutely. Emily.
Sure. Let me try to share. Hopefully this shows up in presenter.
Yes, I can see it.
Yeah, so I'm
just to note that I'm seeing the URL kind of search bar. It looks great, but I might not be presenting.
Yeah, sorry. We don't use Zoom as often, so I'm not sure.
This looks fine too.
Good enough. I won't take up the time to kind of fix that.
You can click on the slideshow at the top. Have you done that?
Well, I did that, which is what is showing up. So
oh, it's fine. It's
otherwise
fine.
okay. Yeah, so I was also going to give another small overview of some physical oceanography work. I'm at PMEL with the EcoFloSci program, and we were looking at mapping thermal habitats across Yukon Chum. And the reason this is focused on Yukon Chum is because they have one of the widest temperature ranges. So one hypothesis was that if the chum migration was impacted by anomalous temperatures, then other species are also impacted. And so this is a model framework that was shared by Joe Langen of the chum migration over the first several years. Then just to show that they tend to spend their winter in the Gulf of Alaska and the summer in Bering Sea, and migrate back and forth through the Aleutian passes. And so this is a framework for developing a model to map that to physical oceanography, and we focused on temperature, which is the dominant impact. And so this is now an overview of temperature for the region for these years, and then I relate that briefly to the chum salmon. This is the black thick line is the climatological seasonal temperature, and then the blue line, I'm mapping the year of the salmon temperatures, and so this is for the Gulf and the Bering Sea regions. And so this maybe relates to the question that was asked earlier. The Bering Sea had high SST in 2019 to 2021, and the Gulf of Alaska was warm in 2018 and 2019, but closer to the mean. And the reason I'm focusing on SST, as um, Bernie pointed out, is that the mixed layer is fairly deep relative to possibly the likely chum feeding depths. So SST is representative of the habitat of chum, particularly in the Gulf of Alaska in the wintertime when the water column is well stirred. But to show that relative to space, focus, for example, on 2022, there were warm anomalies to the west and relatively cooler anomalies to the east. So in the average, the temperatures were closer to mean, but there was some spatial variability. So it's important to map both the temperature variability over time and space, which was the goal of developing this model framework. So a first look is the thermal, taking a binary estimate of the thermal habitat for chum as a boundary, and this is to show that in the winter period, the green shaded areas for each year, it's typically too cold in the Bering Sea, and so the chum are primarily in the Gulf of Alaska, and then they migrate northward in the summer, and as the Gulf of Alaska warms, and in the late summer and early fall, the Gulf of Alaska is generally too warm, and so they are in the Bering Sea, and then they migrate southward in the winter. So this is just a visualization of that migration mapped to these year-to-year variabilities in temperature. And the next level of that model, I took a radial temperature from the Aleutian Islands. And so the vertical black line on each plot is the Aleutian Islands, and to the left is radial distance into the Bering Sea, and to the right is radial distance into the Gulf of Alaska. And it's shaded by temperature, so it's warmer to the south or to the right on this plot. And the gray and black is again a binary model for where the chum would be. And so in the beginning of the year, near the bottom, um, most of the Bering Sea is gray, and so they're in the Gulf of Alaska. And then you can see a thermal migration corridor through the spring, where now the Gulf of Alaska is grayed out. And in the summer, the habitat is in the Bering Sea. And that is in the climatology to the left. And then the year-to-year variability is shown in the other panels. I apologize for the size of the text. One takeaway from this is that in several of the years it was warm enough that there was not great thermal habitat, even in parts of the Bering Sea in the summer, and that there was a large degree of year-to-year variability depending on warm and cold years. And I also did use some climate models to look at that in projections. And I just want to bring up one discussion point is that this work for chum specifically is in preparation. We have an outline, but I want to welcome any co-authors from the project. We've related it to some of the chum catch, but if there's anomalous conditions or habitats that were identified in any of your sampling or research, and you want to try to link it to the thermal habitat, then I'm happy to bring in other partners to pull together some other anecdotes and hypothesis test cases for this work. And I want to transition briefly to another kind of physical oceanography overview, and that also ties back to Bernie's work, is looking at the strength of the Alaska gyre and trends in the occurrence of shelf eddies, just to put the year of the salmon time period in context. This is a time series in the strength of the Alaska stream from sea surface height anomalies, which have been validated by previous moorings in our group. And this shows that in the last 10 years, from about 2013, there's a decrease in strength of the Alaska stream. And for the time period of the year of the salmon project, it was low. And the vertical red bars are also indicating some large shelf eddies. And the distribution of shelf eddies was also low during this time, except for in 2022, where we can see there was a weak Alaska stream and some large eddies present. And I think the impacts of this, I believe Cal is going to talk a little bit about some of the implications of eddies for cross-shelf transport, and Bernie's covered that too. And so I'm just going to leave it there and provide this information for context. This work is also in preparation, but I welcome any other questions and feedback, probably offline or in the chat, just for the sake of time.
Thanks so much, Emily. You're packing a lot there at the relatively last minute too. If you have any questions, feel free to direct them to Emily via the chat. Maybe I'll just leave a couple seconds in case a hand up comes. There's some pretty rich content you presented there. Thanks, Gennady. Okay, if you can answer that one by the chat, Emily, I think we'll move on to, was it Calvin going to be next or Richard?
Richard.
Great, okay. Richard, take it away.
Hi, hopefully you can hear me. I'll just go to share my screen here.
I can. Much better than last time we met.
Yeah, I made sure. So I'll just give a brief chat here. I was looking at kind of a niche topic, which is small organic molecules in seawater, which can form bonds with dissolved copper. The implication is that these small molecules are responsible for modulating the ability of phytoplankton to either uptake or prevent the uptake of copper, which is itself a nutrient and a toxin at the same time. So it's kind of a dose regulating factor, if you want to consider it that. So it's related to primary productivity. All in all, I received about 200 1-liter filtered seawater samples, which I analyzed using a chromatographic method, which I developed. And using this, we were able to quantitate the amount of these organic ligands that bind copper in seawater. In the IYS particular survey, which was more in kind of that winter kind of milieu of the ocean, we did observe a high amount of heterogeneity in our samples, and I would attribute this to the relatively low productivity at this time of sampling relative to other cruises that we've sampled on. In addition, I imagine that what we're capturing is largely more the recalcitrant ligands that are long-lived, and rather than kind of short-lived produced in situ ligands that we might observe in more productive times of year. So in addition to the 201 liter IOS samples, which were from a range of depths from the surface down to 150 meters, I've also analyzed a few hundred samples along the line P transect from 2020, 2021, 2022. And I'll be publishing that hopefully towards the end of this year, the beginning of next year, along with the IYS data that we have, and kind of looking at these ligand values in terms of their geographic distribution, their depth distribution, and the correlation between ligand content and biological parameters, dissolved oxygen, nutrients, dissolved copper, where we have that data available, in situ fluorescence and bottle fluorescence, and so on. At the end of the day, what we have in our lab now is a catalog of extracts that have high concentrations of these ligands, which I've isolated from the bulk seawater. And we're currently working with collaborators to perform more in-depth chemical interrogations of the chemical composition, the characteristics of these captured ligands. So we're looking forward to getting some more chemical data out of this, and as I said, hopefully going to incorporate the data from this initial analysis into a manuscript within the next few months to submit for publication. That's about all I'd like to talk about today, and I'll yield my time over to Calvin.
Hey, Richard, I would expect that you'd have more ligands in regions where you have lots of runoff, or in seasons when you have lots of runoff. Is that true?
Yeah, absolutely. Generally, we have pretty high concentrations of ligands in coastal samples. And looking at the spectral data from these extracts, what we see is kind of a more tendency for these ligands to have more character associated with terrigenous material as opposed to phytoplankton exudates. So it's something we need to explore in more detail, and I look forward to kind of putting more nuance into this discussion with our upcoming paper.
Yeah, also in maybe some eddies that have captured some of the coastal water. That might be interesting as well.
Yeah, totally. We're going to have some chats with our physical oceanographers on deck and get that sorted out.
Thanks, Calvin. Thanks, Richard. Any other questions for Richard before we move on to Calvin? So we've got another question for Emily in the chat. Richard, quick question for you before we go to Calvin. I know you attended a conference early this year in Spain or Portugal on ocean chemistry. Just wondering if there was any kind of takeaways or lessons learned, or even interest from others on this project that would be of interest for this audience.
Yeah, I'd say what comes to mind immediately is there was a new working group founded at the meeting, and that working group is looking at aerosol deposition of trace metals. There was a lot about DOM and a lot about trace metals at the meeting, but definitely this working group is something to look at in terms of they want to conduct some intercalibration experiments between countries to get a solid handle on where metals are coming from in the ocean, how they're getting deposited, and specifically what kind of chemical forms the metals, iron and copper and so on, are taking. Because there's a big question of we have, for instance, aerosolized dust being transported onto the surface layer of the ocean, but does the iron contained in that dust, for instance, have bioactivity, or is it instead just going to precipitate and sink? So there's a lot to be thought about in terms of aerosol deposition and trace metals in the surface ocean in general.
Thanks, Richard. And what does DOM stand for?
Dissolved organic matter. So just any organic chemicals that don't go through a filter, that don't get stuck in a filter.
Okay, I'll hand it over to Calvin, and feel free to post any questions for Richard in the chat.
Okay, I'm assuming you can see this all right?
Yes, I can.
So we'll I'll just have a few slides here. I won't take too much time. Look at nutrient variability in the Gulf of Alaska. If I can figure out how to advance my slides. Oh man. So we had the International Unit of Sampling Stations on the left, and on the right are some historical data that we have. The gray is the ecofoci data along the coast, and then these other lines are historical lines that are collected from this program called GLODAP. So I'll be talking a lot about the eastern Gulf of Alaska. So a nice image of the Gulf of Alaska. We see a lot of coastal regions that have a lot of iron, and then iron rich on the coast, and then iron poor out in the basins. We have generally low chlorophyll, but as we've seen in this talk, there are eddies that foster cross shelf exchange, and so you do get some accelerated additional production out in the basin. But in general, iron is limiting out here, and diatoms are needing of iron as well. So we have a kind of a diatom limited region out here. So here's some climatology from Al Herman. It was and Ken Coyle, Al Herman, and showing from 2000 to 2013. You can see the high iron along the coast in this model, and then especially later in the season where you're getting a lot of runoff and low iron out in the basin. And so what does that mean? Here's a study from Suzanne Strom, which she collected nutrients and phytoplankton community structure at several locations. There's an outer shelf location here, a middle shelf, inner shelf, and in Prince William Sound. And contrasting Prince William Sound and the outer shelf, you can see that there's a kind of a drawdown of one-to-one drawdown of nitrate and silicic acid in Prince William Sound, where you have a lot of diatoms growing. Diatoms, of course, need silicic acid. So out in the basin, and not in the basin, the outer shelf, there were not any diatoms present, or very sparse diatoms. So you're not really bringing down the silicic acid. So here's an example of where the phytoplankton community structure influences the nutrient ratio. So we have a lot of residual silicic acid in the outer shelf, where we don't have the diatoms. So what does that look like for in the eastern Gulf of Alaska? So here's data from GLODAP and the Franklin cruise in the upper 25 meters from February, May 84 to 22, and you can see that nice nitrate silica relationship that kind of bends here, and you do get a residual silicic acid. Now later in the season, when you get a lot of runoff, you can have more silica washing out from onto the coast. So this holds for early in the season, maybe not so much for later in the season. Here's the plots for temperature salinity that were shown earlier, where we have the fresher water and fresher water in the east, and then you can see there's higher salinity out towards the west, and nutrients are salts, so you expect to see higher salts out, higher nutrients out here associated with that higher salinity. So higher nitrate in the west and higher silicic acid. Now there are some offsets between the different groups in salinity and in nutrients, so we still need to work we still need to work through that. But we can see, for instance, this relationship between salinity and silica. So this is the deeper water here, and then with a lot of variability when you get into the surface water, because the nutrients are non-conservative. Well, salinity and nutrients and salinity are more non-conservative at the surface. So here's interannual variability of nutrients in this region near Papa and line P. There's some other historical data along here so this is nitrate versus year here on the left, and silicic acid versus year on the right. So a lot of year-to-year variability, and the red here is out at Station Papa, and the blue is coastal. So you see that near the coast you have lower concentrations of nutrients, and then out near in the basin, then you get these higher concentrations. And then you have a lot of year-to-year variability. The maximum variability we saw in the surface water out near Papa was about 7 micromolar nitrate per 11 micromolar silicic acid. And this looks kind of funny, but if you take, if you just plot up a plot of the data at Papa, there is a significant trend there, probably just driven by these few data points, but it is significant decrease in nutrients. What does that look like in the model data here from Al Herman at Papa? So here's the measured temperatures. Here's the model temperatures, and then he has modeled nitrate. I've included a profile, 2014 profile at along line P with the dark one. The darkest profile here is at Papa. And I just want to point out that you can see a change in mixed layer depth here. Certainly in the deeper water, you can see a lot of variability in nitrate. It's a little higher than what we see at 200 meters in our profile. But you can see also in the surface here that we have, it's getting bluer and bluer. We're getting less and less nitrate in this upper water column. So in the upper mixed layer at Papa. So, you know, we'll have to pay attention to these trends and see how they pan out from the compared to the model. So I think that's all I have.
Thanks, Kelvin. We've got a question from Skip in the chat. Are there any, perhaps for the sake of time, I'll let Kelvin answer that in the chat, and we might take a 5-minute break here for the sake of staying on schedule. So we'll come back in about 6 minutes at 6:15 p.m. Pacific time. And we will be returning with presentation from Marta Koenig on primary productivity. I'll see you in about 6 minutes. Thanks, everyone. Okay, hello again, everyone. We had a good break there. Sorry for making it a little shorter than scheduled. Just wanting to respect folks' time here. So I'm going to hand it over to Marta, speaking on the topic of primary productivity and remote sensing. Go ahead.
Can you see my screen?
Yes.
Good. I will disappear from the screen, but I want to make sure that everything works well. Hello, everyone. After the break, now I will show you the results of the bioregionalization of the subarctic Pacific. All right. The main goal of my research was to identify regions associated with distinct phytoplankton phenology and composition. It's important because, as Cushing hypothesized, he linked the recruitment and survival of salmon to the overlap between phytoplankton and zooplankton biomass overlap in time. So I focused especially on the phytoplankton part and the phenology and timing to describe the kind of the first level of foraging conditions for salmon. Today I will show briefly the workflow of the project. I will discuss the input data sets that relate a little bit more to the IYS sampling campaign. Then I will show you the defined bioregions and roughly summarize the outcomes of the temporal interannual analysis that I did on my bioregions. So first I had to estimate phytoplankton functional types at the stations based on the HPLC data sets and the data collected during the IYS, but also I extended my data set for the data obtained from the open access databases. Then I developed the algorithms to retrieve phytoplankton functional types from the satellite data from the globe color. And also I analyzed and identified the phenology metrics. And my phenology time series and phytoplankton functional types were altogether input data sets for the bioregionalization. So first, the at station phytoplankton functional types that I derived using Kimtax analysis. These are data from the IYS 2022. In the upper row, you can see cyanobacteria. On the left, there is a scatter plot showing Synechococcus counts from the flow cytometry collected during the IYS that I use here for the validation, and they're compared with the cyanobacteria chlorophyll A on the x-axis that I derived from the using the Kimtax analysis. We can see that the correlation looks pretty well. Then in the middle row, you can see the spatial distribution of the cyanobacteria chlorophyll A, and on the right, from the flow cytometry, Synechococcus biomass. In the lower row, cryptophytes. Again, on the y-axis, on the left bottom scatter plot, flow cytometry counts, and on the x-axis, cryptophyte chlorophyll A, retrieved from Kimtax. Then in the middle row, surface distribution of the cryptophytes, and on the right, their biomass. I had only the two groups from the flow cytometry. Those were the main dominant cells that I derived from the flow cytometry, but they agreed pretty well. So here are all the groups derived for the IYS 2022 from the Kimtax. We can see that especially pelagophytes, haptophytes, and chlorophytes group in the southern part. In the northern, I observed more cyanobacteria, diatoms mostly in the areas where Calvin showed the highest nutrient and silicate concentrations. I developed in the next step the satellite algorithms. They worked pretty well, especially for diatoms and dinoflagellates. I had a little bit more problems with the haptophytes, but overall, it was I used all the algorithms to derive the climatology values that I used in the bioregionalization. Here are the climatological distributions, climatology estimated for the years 2002 to 2022. You can see that especially diatoms and cryptophytes, dinoflagellates aggregate especially around, along the margin of the Pacific. Then green algae and haptophytes, pelagophytes are slightly more evenly distributed across the North Pacific. And when it comes to phenology, I used the globe color data set. In the top left corner, you can see validation data set that I used, mostly open access databases. I validated first the globe color data. You can see the scatter plot in the bottom left corner. And today I would like to focus especially on the three main phenology indices. First, the total bloom length, which is the total yearly length of all blooms, the total count of their days. Then the bloom magnitude, which is the accumulated sum of the chlorophyll values within the bloom periods. And then the chlorophyll maximum, the highest value for the entire year. Here is the final bioregionalization. You can see this, especially the bioregions along the margins. I call it even like the marginal bioregion, which is in blue. Then the transitional one and the central part. Here is the shortly characteristics. Central South bioregion, which is mostly influenced by the North Pacific transition zone. Central North, which is the second with the second lowest chlorophyll A concentration and clearly full bloom pattern. Then the transitional zone and the marginal one, where I observed the highest diatom concentrations and the prominent spring bloom. The areas close to the archipelagos, to the Aleutians and Kuril Islands, clustered together. There are also observed diatoms in significant amounts, but there's slightly different phenology with a little bit later blooms, especially southwest of the Aleutians. Up to this part, all the results were submitted to the Progress in Physical Oceanography. We are waiting for the reviews. Hopefully these results will be published soon. And recently I've been working on the interannual analysis, and I was trying to see the long-term patterns and the drivers of the change of the phenology. So for the analysis of the drivers, I divided my bioregions into eastern and western part. So I have marginal east and west and transitional east and west bioregions, because especially in the western part, there is the additional influence of the waters coming along the Kamchatka Peninsula and the mixing with both Bering Sea and the Okhotsk Sea, which changes a little bit the dynamics, and the drivers may be a little bit different than in the Gulf of Alaska. I would like to especially talk about the change point analysis. Here is the total bloom length, and I would like to draw your attention to the shorter blooms after 2014, especially in the marginal east bioregion and transitional east, which is Gulf of Alaska. So after 2014, the blooms were shorter. And the second thing is that I didn't observe any specific trends, but in the years between 2014 and 2021, I saw significantly lower maximum chlorophyll concentrations in these years. This is specifically interesting in the context of the previous presentations. I tried to look a little bit for the reasons. I tried to look for the reasons, and I would try to associate it with the, I checked the mixed layer depth, which was higher But it's interesting to see Emily Lamangi's presentation that the eddy activity was less intense in the last years, more or less, starting in 2018. The other thing is that I've seen smaller diatom contribution, especially again in the Gulf of Alaska, particularly after 2018. But the decreasing trend I've been observing in all the bioregions. And it partly overlaps with the negative NPGO indices, the NPGO index, which has been negative mostly after 2014. But it would be interesting to also compare with the eddy activity. And I think that's a very interesting thing that I've seen today. And it seems that I do really see these changes. Also, after 2014, what Calvin presented, the smaller nutrient concentrations, it seems that we can really see it in the phenology as well. Chlorophyll concentrations are lower, and the diatom contribution decreased in the last years. So to summarize, I defined 5 main bioregions based on the phytoplankton phenology and composition. And especially after 2014, I observed a decrease in the bloom time, especially in the Gulf of Alaska, and a decreasing trend in diatom chlorophyll A contribution compared to the small algae and to the dinoflagellates. The decrease was particularly pronounced in the Gulf of Alaska and the marginal east and transitional east bioregions. Thank you.
Thank you very much, Marta. Any questions? Marta, would you be able to bring up the bioregionalization map again, please?
Yes, of course.
Thanks. Skip, I'm just kind of thinking back to your last presentation on the historical high seas data, and wondering if you see any similarities in patterns of the distribution of catches in these bioregions that Marta has characterized here.
Well, yeah. My talk's at 7 o'clock and we can compare.
All righty. We'll keep this one in the bank as well.
Would you
Any
like me
other
to send you the presentation?
Feel free to send it through to me, Marta. I can pass it on to Skip as well.
Okay.
Any other questions for Marta? Calvin, I'm just wanting to check that you saw the questions from Skip and Gennady in the chat for your presentation.
Yeah, I'm plotting some things right now.
I thought you might have been. I just wanted to check. Great. Okay. I was just about to ask Joanne if she had any similar reflections on the distribution of zooplankton versus the bioregions here, and then I realized she's presenting next. So perhaps the best thing to do is to keep moving towards Joanne's presentations on presentation on zooplankton, and if any questions for Marta come up, feel free to pop them in the chat. Thanks very much, Marta.
Do you see my slide?
Yes.
Okay, great. So I've been working with the zooplankton abundance data from the Bongo sampling. So during the last workshop, I described spatial variation in zooplankton abundance and zooplankton clustering. So I'll just review that briefly. So clustering of zooplankton genus abundances resulted in 8 zooplankton clusters, and we grouped these into warm and cool clusters based on species composition and sea surface temperature. So in the map, the cool clusters are blue and the warm clusters are salmon colored. So the warm clusters occurred in the south and southeast, showing that similar northwest to southeast gradient that we could see in Bernie's work and also in Marta's. So here I'm showing the abundance distributions of clausocalanus and mesocalanus copepods here. So these are the most abundant warm water copepods that were sampled. And they're associated with the transition zone, which extends across the Pacific. So with the cooler water clusters, there were some subarctic taxa that were restricted to the cooler waters, such as the mesopelagic copepod Paraucata elongata here. But most occurred throughout the study area, like Neocalanus cristatus shown here. So the distribution of the warm and cool communities follows general circulation patterns for the regions, with the cool clusters representing the Alaska Gyre and warm clusters showing the influence of the transitional domain, and also the bifurcation zone, where we also see the influence of the California current. So the degree of variation in the latitude of the bifurcation has been noted previously. So our next question was, do we see any differences in biomass between these cool and warm communities? So I reconstructed biomass from abundance, primarily using species and stage-specific dry weights compiled by Moira Galbraith and the folks at Fisheries and Oceans. So these results are still preliminary. And there can be substantial variation in size within species and within developmental stages that result from the environment, and in particular with respect to variation in food availability and temperature. And so this variation isn't captured by reconstructed biomass. And I'll also note that most of the cnidarian data were actually removed from this data set because I couldn't standardize them between vessels, and all ctenophore data were removed. So the biomass shown here isn't the whole community that was sampled, and gelatinous biomass is likely underestimated. So I think that comparing these biomass estimates to measured biomass will be an important step in validating these results. But taking a look at the biomass, we see higher biomass in 2020 and also in 2022 in the area where the Northwest Explorer samples were. And of course, those were collected at a later date. And it kind of highlights that we need to consider confounding variables when we make comparisons between the warm and the cool water communities. So this plot shows the increase in reconstructed biomass per square meter with day of the year. So our models need to include day of year as a covariate, and the analyses that follow have been restricted to winter sampling dates. So this excluded all of the Northwest Explorer samples and many of the Pacific Legacy samples from 2020. And so you can see what this looks like in the map here. So when we compared biomass between warm and cool cluster samples, mean biomass was higher in the warm cluster samples after controlling for day of the year. And so the next sample was, so what's contributing to that higher biomass? So to address this question, we separated biomass into size bins based on species and developmental stage. So which size bin the various taxa was placed in was guided by the Franklin samples, which included size information, and also the Kaganovsky and Pacific Legacy samples, which were size fractionated. So the size fractions used were less than 5 millimeters, 5 to 10 millimeters, and greater than 10 millimeters. And you can see that the higher biomass estimates from the warm cluster samples was due to an increase in the smallest biomass in the smallest size class. And so when we look at biomass and we divide it into broad taxonomic groups, we can see that this results from higher biomass of small copepods and appendicularians. So we don't see a difference in biomass at the largest size classes, but we do see some difference in composition of that biomass. And so I'm just going to highlight one result here, just because this work is still ongoing. But we did see a higher biomass of euphausids in the cooler water samples in the greater than 10 millimeter size bin after controlling for diel phase. So that's just a quick recap of what we've been working on. And in summary, so the warm and cool communities that were identified reflect general circulation in the region. The winter species composition suggests that waters influenced by the transitional domain, the California current, may have higher zooplankton biomass, but in our data, this is attributable to smaller organisms. And at larger size classes, preliminary results suggest a difference in biomass composition that could be relevant to higher trophic levels. Thank you.
Thank you very much, Joanne.
Where's my, oh, there, didn't start my video. Oops.
I see we have Alexei Pinchuk on the call, and just wondering, Alexei, if you have any sort of comments, reflections on what Joanne's presented here, or any things to add from the work that you've done on zooplankton on this project. Are you there, Alexei?
Oh, I'm sorry. I was hi. Yeah.
No worries.
Yeah. I started to talk and I didn't realize I was muted. So yes, I don't have much comments because I've been in contact with Joanne for most of the time when she worked, and we discussed all these findings. And no, I think it pretty much shows what I would expect to see. So
Great, thanks. Any other questions for Joanne before we move on to our next presentation? Okay, feel free to pop them in the chat. I'm sure Joanne will be able to get back to you. Tommy, I see you're primed and ready. Great to see you. I'll let you take it away.
Hello, Aiden. Good to see you. Can you hear me?
Still can.
Great. Thanks a lot for pulling this off. I'll go ahead and share my screen.
Nice toque there.
Thanks.
I
Representing.
should have been wearing mine as well.
Yeah, it's cold here in Cordova. I can get away with it. So can you see my slide?
I can.
Great. Thank you very much. Thank you for the opportunity to present here. I'm really excited to present again on some cool stuff that we did as part of the Northwest Explorers research cruise in spring of 2022, and engagement that we had with some pretty extraordinary programs and teacher and students in Cordova and the Copper River Watershed Project. So my name is Tommy Sheridan. I work for University of Alaska Fairbanks Alaska Blue Economy Center, and I'm really happy to have Kate Morse here with me, who works for the Copper River Watershed Project, and Jennifer Hodges, who's a teacher with Kenny Lake School in the Upper Copper River Watershed. I've presented on this at the June symposium. Kate and I presented on a poster at the symposium in October of 2022 in Vancouver. So I won't spend a whole lot of time introducing what we did. I will be chiming in chat with hyperlinks, but this is a picture of myself and the research crew on the back deck of the Northwest Explorer in April of 2022 on a lovely blustery day in the North Pacific as we were doing some sampling. I was participating in the cruise as a commissioner with NPAFC, and I'm just so very grateful for the opportunity to be a part of this and to share with you all what we did here today. So when I got on the boat, I didn't really know what to expect. I'd been on research cruises and, inside waters of Southeast Alaska and Prince William Sound, but I'd never been out on anything like the Northwest Explorer or a part of anything like International Year of the Salmon. Didn't know if we'd have connectivity or communications, and I was really happy to find out that we did have really reliable telephone communication. And so I was able to connect immediately with just some really innovative folks, Kate Morse and Jennifer Hodges, and I'm happy to introduce them here to you all today. I was able to, as a part of our work, connect with Jennifer, who's in the picture here, and her students at the Kenny Lake School, just to talk about International Year of the Salmon, open ocean research, and as you'll see, I hope, if you're able to follow all of the hyperlinks that I share with you all, just how engaged she and her students were with International Year of the Salmon, and then otherwise just the real innovative engagement with salmon biology and salmon fishery management that Jennifer and her students in the Copper River Watershed Project do as a part of their work. So that will be the end of my introduction. I'm really happy to have Kate here, and so I'm going to hand it off to Kate. I'm in Cordova, Kate's in Cordova, so hopefully our bandwidth cooperates, and then Jennifer will join us from Anchorage here shortly. So Kate, can we hear you?
I hope so. Can
Great.
you?
Yep.
Great.
Take it away.
All right. Well, good evening, everybody. Just a brief background about our organization. So we're a nonprofit, like Tommy said, that's located in Cordova, but we span our region of focus is the whole Copper River watershed. And so for now 25 years, we've been working on our mission to promote a salmon-rich, intact watershed and culturally diverse communities by forming partnerships for watershed scale planning and projects. And so that includes working on ecosystem monitoring throughout the watershed. It includes working, implementing active restoration. One of our banner efforts has been removing fish passage barriers. And we just completed our first bridge, opening 70 miles of upstream rearing and spawning habitat for king and coho salmon. And then education is another important one because the long-term health and the long-term sustainability of the watershed and these restoration and monitoring efforts that we do don't mean anything if there's not another generation to carry it forward. And so I just feel very honored to represent. I know that there are many organizations out there that exist that are taking a watershed approach. And so I'm just one of them and would like to use this opportunity to just advocate for the importance of nonprofit partners in taking the wonderful work that you all are doing, especially out in the ocean, and connecting it to the freshwater landscape, because what happens in the ocean is going to affect what's returning to the streams, and then what makes it to the ocean is also affected by that, as you know. So I think that we play an important role. I might not be a specialist in the chemistry or ecology. I consider myself a generalist with my biology background. But what we as nonprofits can be is flexible, creative. We work to convene and bring together expertise. We can pursue resources and just help connect the science to the people, help engage the people in the science to be able to reach and gather data in places that not every scientist can get to as easily as some of the residents who live, hunt, recreate in these areas. And then from the education side, I just want to emphasize the importance of connecting kids, whether it's throughout different ocean communities and the coastlines, whether it's in our case throughout the watershed, any chance that we can to connect students that live in disparate communities that seem very far apart from each other, but are connected by the waterways, is an opportunity for them to look beyond their neighborhood and their school and their immediate community and see that they are an active resident of a whole watershed community in our case. And it's like I mentioned earlier, we've been doing this for 25 years, and those relationships don't happen overnight. They've been building for 25 years. And the other point I would like to emphasize is and this came up in our presentation that we originally gave, but in this poster that you see on your screen, we included a ladder because we don't have a full-time education staff that can put on programs for every grade, every student all the time. But we've developed a model where we can reach students at multiple times with our partnerships at multiple times throughout their school career, so that each lesson and experience builds on the previous, so that by the time that they graduate, they have a very strong sense of belonging to a much bigger ecosystem, and having a responsibility for the stewardship of that ecosystem. And I can say with confidence that it's working. I've been in our organization for 15 of those 25 years. And this summer, 2 of our former students were on the payroll, one of them a college student, the other one a high school student, that have been a part of our programs at multiple points and have come back to help explore careers that are actively involved in stewardship of the landscape. And if that's not enough evidence, I'm super excited that Jennifer, one of the amazing teachers that I get to work with, is also able to participate tonight and can give you a report from the classroom perspective of what these experiences and opportunities mean for students. So to you now, Jennifer.
Thank you. So I got interested in the Salmon in the Classroom project because salmon is a very big resource here in the Copper River, especially with my kids who are right on the edge. That they do dip netting and fishing and subsistence, and but that's really all that they knew about salmon. And in education, or in elementary school, I should say, we do everything off of a driving question. And our driving question was, what do you know about salmon? And I just was just really shocked that that's really all that they knew about was subsistence and tourism and dip netting. And hey, we've got salmon in our fridge. And that was about it. And so along with Kate Morris, a lot of help from her, we have really went on a journey. And the life cycle has been a big thing in the classroom. Of course, they follow the journey, the unique journey of salmon, and now estuary is in their vocabulary, where a few years ago it wasn't, and that's pretty impressive for 8-year-olds. And they get involved with ATU data collecting. They get really passionate about the subject, and so much so that they've made YouTube videos. We've went to the hatchery. We've been published in an online curriculum with McGraw-Hill, featured on NPR, and that's been them. It's just their excitement that they get around learning more. And so the culminating big thing for them is the end of the year when they get to release the salmon, and it's just exciting for them. They get to observe the habitat, they get to collect data, and Kate is right there getting them in their chest waders and getting them out into the lake, and they're looking at what are they going to eat, what's down there, and it's just really exciting. But they've also had some really good interactions with the salmon as far as failures. One big one was right after COVID-19, where hand sanitizer was a big thing. Spraying down the classroom was a big thing. And they saw that just introducing those few little pollutants really built up. And by the end of the school year, we had a very high mortality. We only had 7 fry out of 200 make it. And we did a lot of samples of our water quality, and it was found that, yeah, it was just this pollutant that you would not believe would just be there just a little bit at a time really made a big difference. And then last year we had another big one, and that is we had a power failure. And within just a few hours, our temperatures the ATUs, or the temperature units, went up and just by a few degrees, and it wiped out the entire tank. And that was pretty devastating to an 8-year-old, to walk in and all of your eyed eggs are gone. And that really powered them up. I mean, they were mad. We had some pretty powerful discussions about climate change and pollution. And to get an 8 to 10-year-old to really have these powerful discussions is pretty amazing. And that's because of the the watershed project, putting that into the hands of teachers, just allowing us to be able to have the salmon in the classroom. So it's been exciting.
Thank you, Jennifer. Thank you, Kate. I'm just so very glad that both Kate and Jennifer were able to join us. I'm gonna run through some slides here real quick, but I'll be dropping hyperlinks into chat, with apologies for any distractions. I'll be providing a hyperlink to Jennifer's website, to the Kenny Lake Hawks YouTube page, so that you can see all of these cool videos of students visiting the Salmon Gulch Fish Hatchery, for example, and other salmon related activities. And then the story that Jennifer made reference to of NPR covering some of what she's doing with her class, I'll provide that as well. So that's really all we wanted to do. Just wanted to put faces and voices to names and experiences, and really just drive home the importance of connecting science with students. It's really important for these communities in coastal Alaska that are so heavily dependent upon salmon for their livelihoods, for their culture, for their subsistence, and just always want to drive home the importance of that for the work that we do. So if anyone needs to get in touch with us, they can do so through my contact information, and really just appreciate the opportunity to share a little bit about what's going on in Cordova and the Copper River watershed. It's a really remarkable place, and could very well be a part of future iterations of International Year of the Salmon, be it Becky or other initiatives in the future. So thank you very much for the opportunity.
Thanks very much, Tommy, Kate, and Jennifer. Yeah, really, really great to see that local, on the ground impacts and then inspiration that projects like this can instill in youth. With my current role with the Council of the Haida Nation, we've been privileged enough to participate in some offshore expeditions as well. While I've been there, as well as in previous years, and there's been incorporation of those, there's been communication between vessels, scientists on vessels and school students in communities in Haida Gwaii, and I've heard very similar positive impacts. So it's great to see, and it's making me think of with Becky moving forward, and how to link that with local, the needs of local communities. That I think this communications and education piece is, perhaps something that could be explored, where often, these remote communities, like you say, Tommy, are those that depend and are so closely linked to salmon and other marine species, and providing opportunities for that educational connection could be a real benefit, like you've displayed there in the Copper River watershed. So in this instance, I think it's been spearheaded by you, Tommy, kind of taking initiative and using your connections to make this happen. But perhaps moving forward, that could be something that is written into the research plans or something like that. But I'm wondering if you have any suggestions on how that could be more made more accessible with, or at least bigger projects, research projects like this, how they could be connected to schools and local community education opportunities like this.
Well, this is one excellent example. in my opinion. And it's likely mirroring opportunities elsewhere throughout the basin. But I think it's a potential opportunity if for example there was benefit or value or interest in selecting locations to kind of focus on these opportunities and these relationships. Cordova, Copper River has kind of a built-in ecosystem, so to speak, whether it's through the Copper River Watershed Project, its engagement with the school districts. The Prince William Sound Science Center is one of many partners in that as well. So I think we're probably just, in my opinion, an extraordinary example of what's out there. But I would expect and hope that there could be identification of strategic partners throughout the basin.
And I'll add, I think it's also important not to make it too complicated. Tommy just offered to use a cool phone that was able to connect from the deep open ocean to a coastal school. It certainly takes the support of an amazing teacher. And so I think that creating either incentives or benefits for participating could, be able to help encourage and I mean, Jennifer doesn't need that, but for some teachers, it does take a little extra work. And so that's, being able to make that accessible from the teacher's perspective as well. But it doesn't need to be complicated. I think that there's a lot of local partners and a lot of, and many communities that could help play that role in connecting. And I think the incentive for the research, from the research side, is not only the connection of your science to the public and including the youth, but in working with Jennifer, the reason that they have a YouTube channel is she and her husband are amazing at pulling together really awesome videos. And that has helped with my work where that's not my skill set. And so now I have a great partner that are helping to produce great outreach materials to promote that work that we do. And so the benefits go both ways. And so I just wanted to add that. Thank you.
Certainly. Thanks, Kate. That's a good reminder that it doesn't take, a huge amount of resources to make it happen. But yeah, just a phone and some enthusiasm is all that's needed. And I'd encourage folks to check out the videos and links that Tommy presented there. I remember watching them, maybe it was in the last workshop, and it's great to see the enthusiasm of young folks. And I think that, with this project, we're very focused on the science element, but inspiring youth and getting youth to be interested in science and care about the environment is a big part of making change and improving sustainability. So something that shouldn't be forgotten, and I'm really glad that you've highlighted that here. Any other questions for Kate, Jennifer, or Tommy? Okay, well, thanks so much for joining us. Really appreciated that presentation, and we're going to move on to Skip McKinnell for his highly anticipated talk to finish off today's proceedings. Take it away, Skip.
Roger that. Let me see if I can get this thing shared. Okay, let's try this. Okay, this talk is not highly anticipated because it was an afterthought. But it was a result of my working a bit with a data set that I've created with Joe Langen's help and many data contributors. But in going through it, I thought I would, I'm not sure how many of you will continue working on the high seas in the future. I'm going to assume that there is a percentage. It might not be a big one, but so this is kind of a message for people who are planning to work in the future. And there are a couple of things that I want to touch on. The first is just the state of what I've called the Pacific salmon data legacy. And I want to just talk a little bit about some gear comparisons that became evident to me, and that I thought people who plan to go to sea again should think about a little. Okay. So the Pacific Salmon Data Legacy was something I started during the COVID era. And my goals were to seek out historical biological data about salmon sampled at sea. So initially it was focused on the INPFC era, which ended in 1992, but there was some mission creep and it got a little bit bigger. My core variables for biological data are species length, weight, sex, age, and gonad weight. Eventually IGF-1 will get there, but it's not there now. The greatest risk, what I thought was most of the biological data for salmon sampled at sea, the individual measurements are hardly ever published. And so if they're not looked after, then they're at, I think, a higher grade of loss than the catches, which are generally published in throughout history. So that was my second goal, was to seek out the historical research catches. And so what this contains now is catch and effort by species by haul for about 125 vessels over a 50-year period. These records were transcribed from source documents or electronic files that people donated, but the goal was to create files with common codes and formats. And the result is there are station/bridge log files, there are catches, there are species and biofiles. And I wrote about almost 16,000 lines of transcription code to get it. There are about 121 different sources at the moment. And they're all linked together by a Hull ID created, which was code which I created, which is, one gear, one hull. And it's VV is the vessel, YYY is the year, DOY is the day of year, and NNN is the event. So here's what's in the PSDL at the moment. 43,668 hauls, of which 80% of these have salmon caught. There are some notable exclusions which are important in interpreting whatever I'm going to show you. Most of the salmon Japan Sea data was excluded, and that was by choice. I don't have much BC data after 2009. Washington data they've been sampling on the West Coast since 98. I have none of that. I have the Russian basis data, but not the non-basis data. And I don't have much Japanese data after 2008. So here you can see on the right, you can see by year, the number of hulls in Eastern and Western hemispheres that are in the database right now. And here it is whether they're off the shelf or on the shelf. You can see there's been a rather dramatic decline in the amount of work done well, maybe I shouldn't say that. In my records, the amount of records I have with basin data diminished markedly after INPFC ended and NPFC started. But I want to announce to you who are interested in high seas salmon data that the first public release of this data set was sent to the Secretariat yesterday. And whatever machinations they have to do to make it available, they will do. And presumably it will become available at some point in time using protocols I assume that Brett Johnson talked about a little earlier. Okay, so here's the shot I wanted to show you let's go back. I wanted to show you this is kind of this plot reminded me of a US presidential election. There are red dots at the top, which represent pixels, 1 degree latitude by 2 degree longitude, where the average CPUE of sockeye is greater than the average CPUE of pink, which are in this periwinkle color on my screen. And this is for the month of April, which was as close as I could get to winter sampling. Because you know, if I'd only put up January to March, I wouldn't be able to fill in as many pixels. But this was a shock to me. I didn't expect it. So that if the CPUE of sockeye was greater than pink, I put it as a red dot. And if the pink salmon was greater than the CPUE was greater than the sockeye, I put it as a periwinkle dot. And this spatial structure, it kind of blew me away. As though there is a Mason-Dixon line you can draw across the North Pacific that separates the pink salmon and the sockeye. But that's exaggerating, of course, because certainly there are pink salmon north of that line and there are sockeye south of that line. But here's how they line up if you just look at which one has the higher average CPU. And this is for the month of April. Thought you might be interested in that. And I've overlaid here these little black squares that came up. I've overlaid those, and they are the 2022 IYS cruises. So these cruise tracks essentially were sent to the part of the North Pacific where sockeye dominate and pink salmon are not dominant. And that in part might explain why why there were no pink salmon or few pink salmon in the catches. You have to work further south, I guess. So I wanted to talk about gear comparisons. And I want to start with French's statement in 1968. They spent a lot of time on gear intercomparisons. And here, there is little difference between gillnets and purse seines in the age and species composition of the salmon caught. But the long lines are selective for the older sockeye and chump salmon. So when you're comparing these, you need to think about what the gear is doing to your samples. And there has been very little effort, in my view, to do an intercalibration of any of these gears with trawl nets. And David Welch tried it in the 1990s and found that he didn't catch anything with his trawl nets on several attempts. And so he switched over to longline and gill net and caught salmon. And you remember that on the Raw Spirit in 2022, Dick Beamish, his cruise caught a species composition that was quite different from what the other vessels were catching. So here's what we've got in the historical database right now for gear use. There are 6 gear types: beam trawl, gill net, floating longline, purse seine, midwater surface trawl that everybody's using now. And there was even vertical long lines attempted in the early 60s. And so you can see on the right, the G for gill net is a lot more effort, a lot more records in the database right now for open ocean rather than shelf use of gill net. Same holds for long line. The trawl samples are the other way around, more on the shelf than in the basin in the records I have. And so if you look at the catch frequencies by gear type, everything is squished over to the low end. And in part, that's because the largest haul on record was over 17,000 salmon caught in one set in 1975. So that tends to skew your distribution curves. If you take the square root, you start getting a hint at some of some differences between the gear types. Certainly, you're seeing gill net, you're starting to see a distribution in long lines, but the other ones are pretty tight against low low CPUEs. So this is one of the plots that I wanted to show you. And you can see in the top left is the beam troll. And I've plotted this by number of species caught. So that's what the number per haul is, number of species caught per haul. And you can see with the beam troll, lots and lots of zeros and pretty low numbers of catches of 1 through 6 species. The gillnet looks kind of normalish, with a mode of about 4 species per haul. Longline is 2 per seine. Anyway, you can see these, but the thing that struck me in looking at this was the zero frequencies on both the trawl types. And whether it's the contemporary midwater trawl or the beam trawl, like 0 is the most common number you get. And that differs from the other gear types. And anyway, so these are restricted to open ocean trawl sets rather than shelf trawl sets. And here's a look at the winter. And in winter, which lasts up till April in this plot, the modal number for the trawl type is still 0, and it's different from the other gear types. So that was something that I thought I would point out. Funny enough that when I compared the basis data from 2002 in the Bering Sea and Aleutian area, 4 years of that, and you don't get this, this 0 predominance in the trawls in the Bering Sea, whether it's Russian or United States fishing. They are both. You probably can't distinguish those 2 distributions. Anyway, so the issue is keep on trawling. And I suppose it depends on the question you're asking. And so in the Gulf of Alaska that I've been showing you, you get fewer species on average. And yet, the fishing times are similar to what you have with longline and purse seine. Sample sizes with trawls are generally smaller. You hardly ever see any age data out of a trawl net. And I really wonder what this excess of 0 catches means for using trawl gear. Does it reflect fewer salmon to catch or higher gear avoidance? Anyway, it was just a question that came to my mind while I was looking at some of these data. So I'll take any questions if you have them.
Thank you, Skip. There's one in the chat from Jacques White. Any gear comparison information for steelhead?
Gear comparison for steel. I mean, it could be done, certainly. There are, as I said, there are 43,000 hauls, and steelhead form. Steelhead is kind of unusual. In the INPFC convention, they were not a focus fish. And so what you often saw for steelhead on the high seas is that they were reported by Canada and the US, but often Japan would not report them in the same way. But when I was going through certainly the 1960s Japanese data, I was able to find, well, first I learned what steelhead looked like in katakana. And then in the remarks column, they would often have the number of steelhead caught, but not identified to the mesh size in the gill nets or something like that. But for a bunch of the cruises, I was able in the 1960s to get steelhead data.
Thanks, Skip. Got a hand up from Cam. Go ahead, Cam.
Thanks for sharing, Skip. Always a pleasure. I had two questions. One, hopefully easy, one much more nebulous. The first was, with trawls, we pretty much always are setting them blind effectively. Like, we make sure that we're not going to run over a seamount and get hung up. But with seines, at least in coastal seines, you're commonly targeting schools or looking for school activity. Do you have any idea if they were, I guess what I'm asking, fishing in substantially different ways when they were using gill nets as opposed to seines, as opposed to trawls? I guess with gill nets, you're just setting and letting them soak. But especially with seines, would they have been using like spotter aircraft or smaller spotter vessels to basically try to identify a place to set as opposed to setting blind?
Well, that's a good question. I remember talking to Vladimir Radchenko about his trips on Japanese vessels, and they would, look for things like the color of the water to deploy their nets. So, a lot of the gill net fishing was done by commercial vessels under contract to the government and of Japan. As a matter of fact, that was the case in Canada as well, and presumably the US. That they had guys who were, I'm sure it was all guys, who were fishing with some knowledge of how to catch salmon. And I'm sure that developed over the years.
Yeah, that makes sense. Usually commercial guys are pretty good at this sort of thing. If there's time, I had another question, which is, I'm getting a thumbs up from Aiden. And it goes back to your democracy in the high seas CPUE plot.
Yeah.
Given this sort of ongoing debate about the importance or unimportance of pink salmon in terms of density dependent processes, what do you think the obvious next steps of sort of best resolving what the mechanistic linkages are, given that we keep on seeing statistical evidence for pink salmon effects, we keep on failing to see really clear evidence of overlapping spatiotemporal distributions? How do we reconcile those two things?
Well, you have to, in my view, you have to think of this as a crime scene, right? The pink salmon are doing nasty things to the sockeye, and we want to catch them in the act. Part of the motivation for the pink versus sockeye, pink interaction with sockeye and reduced growth arose from using the species ranges as the argument for overlap. In the days those were the only information you had really about oceanic distributions. And as a consequence, you have to remember that politically, it was really important for people to show that their salmon covered the most of the Pacific that is possible to claim as your own, because it was talking, the main themes then was how are fish intermingling in the open ocean. And so the diagrams of the era tended to show large species ranges, whether there was any density of fish in there at all. If you found a tagged fish that was from your country at some remote place, you were allowed to draw a line way out into the ocean and claim it as a place where your salmon went. And politicians, I think, were happy with that. But now I think this is the first time that with this, as comprehensive a data set as we can get, I think we can start looking, as Joe Langan has done, at how these essentially probability density profiles of species in time overlap. And I think that would be the first really good thing to document, would be that. Let's move this away from overlap of species ranges to looking at densities in time and space and how they play out. So that's what I would suggest.
Thanks, Kip. Thanks, Cam. I'd like to put a question to Dick Beamish, who I see is in attendance. Dick, you were behind the raw spirit and utilizing gill nets and long lines there, and you probably had some time to look and foment over that data over the last year or so. What are your thoughts on the next steps here, and any comments on Skip's presentation?
How much time do we have?
10 minutes.
Well, first of all, it's important to do what Skip has done, but there's a complexity there. Skip, I understand, has not really included a lot of the Russian data, and the Russian data is extensive. And using trawls was an intent to estimate abundance. That was the that was the overall intent of the Russians. Going back to the purse seine data, if I remember correctly, a lot of that was the intent was to tag fish. And so the paper that came out recently on grouping, very quickly, I went back and looked at the data that were available for that paper. And it is extremely difficult to match up the data that are included in that paper with the data that were collected. And that may just be my problem. So, and then the comment about the overlap of pink and sockeye. Well, if you go back to the catches in the 1960s that were all the high seas tagging, in the winter, it was pretty clear that pink salmon tended to be south in the winter. At least it was clear to me, I think. And pink salmon still tend to move south. Do they move south because of temperature, or do they move south because of latitude? All of this meaning that with the changes that we're seeing, that we all collectively want to really step back and rethink about some of the relationships that we normally accept as being reliable. So the problem with seeing the apparent interaction between pink and other species, are there other explanations? Could the explanations relate to production, say, of squid salmon? There is some evidence that there is odd and even year productions of squid. So you asked for my comment. My comment in general is that all of us scientifically want to think about what it is that we really need to know. In a very general way, the consequence of the warming that we're seeing now requires that scientifically we rethink a lot of the relationships that we traditionally accepted. And that's a little bit, I think, what Skip was saying, that go back to the reason that we use trawl nets to fish for salmon is because we want to comment on abundance. And so you have to have a stratified sampling program, and you don't catch salmon sometimes. So I didn't really come up with a definitive answer to anything other than the changes that are going on now with salmon throughout the North Pacific indicate that scientifically we all have to step back and rethink what we think is traditionally controls salmon abundance, which is why we had the International Year of the Salmon, The original intent was to get teams of researchers from all salmon producing countries to work cooperatively to demonstrate that we could do that. And certainly I would like to see what I call stream to stream studies in which you look at the whole life cycle of Pacific salmon. And I don't think that we have done that, but you need to do that. You need to do that on an international scale. Did I take 10 minutes?
That's 5. That's great, Dick.
Okay.
Dick, I know that I think we're planning or trying to plan additional expeditions after the 2022 expedition.
Say that
Yeah,
again?
and this is Calvin. I thought Emily's presentation was approaching a stream-to-stream analysis. I thought that was a pretty neat approach. Anyway.
Yes, but, and of course, and if you had a stream-to-stream approach, most researchers would continue doing what they're doing, but in addition, you'd have to add some new science. So in a sense, it's supportive of what's going on, but it recognizes that there's more value to the work that we're doing if you actually could add the nearshore studies and the open ocean studies, and particularly studies when salmon are returning to coastal areas. I think we really have to do that research, and you want to do that with international teams of researchers, and that's what IYS was all about. Oh, sorry, that's what I thought IYS was going to be about.
Thanks, Dick. Thanks, Calvin. Dick, I was just asking, do you have any plans to organize further expeditions?
Who did you ask?
I asked you, Dick.
I had a proposal to do one this year, and it was to be essentially a purse seine study, which would have been off BC, Washington, and Oregon, with a focus really on pink salmon and coho. And I had worked with a person named Brian Kinzit, and we had what I thought was strong political support. It was going to be run by First Nations in Canada and tribes in the United States. And I called it an international study of the winter ecology of salmon. And then just out of the blue, the people that reviewed our proposal didn't think that we had the ability to carry it out. So I don't know what happened. I then tried to find out if there was any support to put in a proposal, not for 2024, but for 2025. And I haven't heard back whether there's any government agency that's interested in that. But I think there's no way that we can do it for 2024, but 2025 will be another odd year pink production, which could be another major production for pink salmon. And it might be, I think it would be important to do it in 2025. There doesn't appear to be any interest that I can hear from government agencies just yet, but I haven't given up for 2025.
Thanks, Dick. I see that we had 2 questions in the chat from Laurie and Gennady, just speaking to the fact that the cameras placed in trawl nets have observed fish escaping or swimming out of the trawl nets. Skip, any comment on that?
Well, if you're catching fewer species, something has to be leaving the net or somehow avoiding it. Yeah, that's it.
Thanks, Skip.
Okay, can I just interrupt for a minute? The Russians, if I remember, have a catch efficiency for their troll nets of about 0.35 or something like that. And they came up with that estimate experimentally. So they accept that, if I remember correctly, about 2/3 of the salmon escape from the trawl catches.
Thanks for sharing that, Dick. I think with that, we are at our time here. So I'm going to wrap today's proceedings, and a big thank you to everyone that's still around and those that presented earlier. We'll be back tomorrow at 4:30 p.m. Pacific time. But thanks very much for those that have joined.